All right, so today we're going to be talking about the Sequora small genome phylogenomics pipeline. This is the premier pipeline on SequoraDNA.com at the moment. And basically what this does is it compares full bacterial genomes to one another, or full prokaryotic genomes. You can use it for bacteria or archaea. I may say bacteria, I may say prokaryote. I mean, tomato, tomato at the end of the day. You guys know the differences between bacteria and archaea, probably, if you are watching this video. So let's, without further ado, let's dive into this, because I think that as I go through the pipeline, things will pretty much be explained, and you know, I can answer things as they come up. And of course, if you have any questions, feel free to leave it in the comments section, and I'm sure either myself or somebody else can answer those questions. So let's take a look here. So right here on the home page, you got the Sequora small genome phylogenomics pipeline. Now this is divided into multiple parts. So here in parts 1 and 2, what we do is we upload our input. So before you even get to parts 1 and 2, what you should do is go on NCBI or go on other databases, obviously, and download the full FASTA files of whatever genomes you want to compare. Now, I'm not going to go into how to do that in this tutorial, because that, I think, is a tutorial for another time. However, once you download those, you can actually merge them using a FASTA file merger that I have available right on Sequora. And that can be used for this pipeline, it can be used for other purposes, and it's actually been a very popular FASTA file merger, which I am very pleased with. So you're going to take those multi-genome FASTA files, you're going to merge them into one file, and you're going to upload this file to parts 1 and 2 as both the query and the reference input. Now, for more advanced users, you might be able to change this up a little later on, but for this tutorial, we're going through a basic workflow. So take this multi-genome merged FASTA file, upload that to the query, and upload that to the reference. And then what you're going to do is you're going to hit this button that says Process FASTA Files, and you're going to be given 3 outputs. Make sure to download all 3, because you're going to be using these later on. So we're going to go ahead and download all 3. And then part 3 is the main workhorse of the Sequora pipeline. So we're going to upload the chunked query file, and we're going to upload the concatenated reference file. Now, to explain this a little bit, what happened in part 1 and part 2 is that the query FASTA files were divided into 1,000 base pair sequences, and the reference files were divided into 10,000 base pair sequences, which were then concatenated to give 20,000 base pair overlaps. And I give a little more detail in the instructions if you want to know a little more. So we're uploading those to part 3 as the file input, and then we're going to take a look at the scoring parameters a little bit. So you can see the defaults are what they are. The worker settings, we have it where the CPU cores detects 20 on my computer, number of workers is 10. So you want the number of workers to be about half of your CPU cores if you're just starting out. As you go along, you can feel free to increase that. Now, pretty much by definition, you shouldn't have more workers than your number of CPUs. However, if you would like to increase them as you go, that's fine. That's something you take a look at. However, if the number of CPU cores you have is more than the number of workers, make sure to adjust that manually. And then, of course, we have the k-mer filter settings. Basically what this is it says, okay, we're going to divide the query sequence and the reference sequences into tiny k-mers of a length of 15 by default. And if you don't have it where the number of query k-mers is found in the reference k-mers at least 30% of the time by default, so so that's minimal k-mer matching, what's going to happen is that the system is going to skip over that one, and it's not going to perform the alignment, and it's going to register those as it's going to register those later on in some of the statistics. So if you have it where this query file cannot find any reference sequences where it passes the k-mer filter, it's going to be registered as unique, and that'll be important for these. I'll explain that a little bit later on. So with the file input, we're going to upload the query FASTA file, upload the reference file as already explained, and then we're going to hit run alignment. Now, this is going to take hours to days, typically, depending on how many genomes you uploaded, depending on the capabilities of your computer. So this is going to take a very long time. Now, you might say, well, I don't have all that time in the world. Well, the good news is this is a good time for a coffee break, or this is a good time to let your computer run, and then you can go home and get this thing that scientists are often not familiar with called sleep. So this will be a good time to take a break from your work, let your computer run overnight. You don't really have to do anything with it, and it can just go ahead and run the files. Now, since this is going to take hours to days anyway, you don't have to be right there babysitting it. And eventually it's going to say that it's completed. And so once you have that, go ahead and download the output, and this is going to be very important later on. So click download CSV. Now, the pause and resume buttons, feel free to use those, but use them somewhat at your own risk. I find that how well they work is a little bit spotty. This is not perfect software. I am not Microsoft. I am just a guy building this using the tools that I have available. So after we download that, we are going to go ahead and move on to part 4. Now, part 4 is divided into part 4A and part 4B. And what this is going to do is this is going to help to organize and process a lot of the results that we saw from part 3. Now, like I said, part 3 is the main workhorse. That's the part that takes hours to days. The rest of this pipeline actually goes pretty quickly and requires very minimal computational input. So we're going to hit process data after we upload part 4A, and then give it a few seconds before you hit download process CSV, just to make sure that everything is properly processed and you don't get something broken. And then you're going to upload the chunked query CSV that you got from part 1 and 2, and you're going to upload the output from part 4A to part 4B. There is a reason this had to be divided into 2 parts, and I'm not going to go into all that detail right now, but it did make it a little easier on the computer to divide this into part 4A and part 4B. Keep in mind that this is a browser-based pipeline, so in order to keep within the memory limits, that is part of why this pipeline had to be divided up the way it was. And this also allows users later on, if they want to do more advanced things with this pipeline, like they don't want any alignments that were to be included that were, say, below 70%, they can go in and manually filter a lot of these using Excel. So each point in the pipeline allows users to go in and play with the data. Honestly, obviously, I'm not suggesting cherry picking, but they can go in and manipulate the data in order to help to answer whatever questions it is they're trying to ask. And I tend to be more team questions than team hypothesis. But obviously, that's something we could get into in another video. So over here in part 5, we're going to upload the outputs from part 4B. We're going to hit process file and give it a few seconds at least, let it process, and then click the buttons for the downloads. And then after you download those, you can go to part 6 to 7, which is the final part of the main pipeline. Now, in part 5, you saw part 5 did a lot of the analysis already. So part 5 will tell you how similar any two genomes are to one another. So here is bacterium A, here is bacterium B, or here's archaea A and archaea B, and we want to know how similar are they to one another. And the reason I did that is a lot of times we'll hear things thrown around. Like we'll hear, humans and chimps are 98% the same. But as Richard Buggs, the evolutionary geneticist over in the UK, explains, actually they're closer to around 85% the same, which opens a lot of other philosophical questions. So what do we mean by how similar are two genomes? So in terms of bacteria, if I want to know how similar two E. coli genomes are to one another, that's a very interesting and very important question, and that might be separate from how closely related they are. So you might have it where two genomes are very closely related, but they might be very different because of perhaps a large genome rearrangement that happened relatively recently. Perhaps there was something involving a plasmid, perhaps there was something involving some type of movable element, and perhaps even events that we haven't even thought of yet. And maybe Sequora will help to uncover some of these. So in part 6 to 7, we're going to look at phylogenomics trees and phylogenomics webs. So we're uploading that output from part 5, and specifically we're going to upload the averages output, as you will see here. So averages data part 5, and there's a lot of variables here. You can go ahead and change these variables however you like. I find that the defaults work pretty well, hence the reason they are the defaults. But if you want to change things around a little bit, you are free to do that. Now, when we hit generate trees, you're actually going to get multiple outputs with a bunch of different definitions of similarity. So you'll get combined metric 1, combined metric 2, and in addition to that, you will see alignments, average alignments with and without gaps. So you get a whole bunch of metrics here all at once. Not just one tree, not just a few trees, you get a whole bunch of trees. So as you scroll through this, you will see a whole bunch of different trees here. And since this is more of a map of similarity, that already opens up a bunch of questions. Of course, what do we mean by similarity? And within these trees, you will see a root. You'll see circular trees. You'll see more traditional rooted trees, even though the circular trees are also rooted. And you'll also see these phylogenomics webs. Now, with these phylogenomics webs, this right here is very much a map of similarity. You have inside of it a circular tree, but then each leaf in this tree is connected to each other. So what I did with this is I wanted to color code, did they overlap? Is there overlap between them? How similar are they? And so there is a very minimal visualization of the data already. And for more details, you can go into the outputs within the spreadsheets. So for example, I am looking at a particular output right now. So I'm seeing combined metric 2, I'm seeing the phylogenomics web. And with that, leaf 1 and leaf 6 are connected to one another using a kind of a purple line. And that purple line means they have overlap with one another. However, 1 and 7, there's no overlap. Hence the reason they are connected with using more of a cyan line. So it means that all the chunks between them were unique. It means none of them passed the k-mer filter. And that affects how similar they are in terms of the calculated metrics for combined metric 1 and combined metric 2. So as promised, we're coming full circle back to that explanation. So this offers a lot of convenience for a lot of reasons. You might say, well, are those two genomes just not related to each other? And you have to consider that the history of life on Earth is not as simple as a slow and gradual change and slow and gradual branching. If you have a plasmid, for example, included in your data set, you might have it where that plasmid overlaps with some genomes and not with others. And that might mean that some of these genomes have that plasmid embedded into them and some of them don't. Or perhaps a part of that genome was taken by the plasmid and it overlaps with some genomes within that clade, within that group of genomes, but it doesn't overlap with others. And so a lot of questions are opened up. And so I wanted to go beyond the assumption that just because we see similarity, that means a branching family tree. Because we all know that bacteria can be very, I'm not sure if I say the P word on YouTube, I'm not sure if that's going to get flagged, so let me be careful with that. But they like to be friendly. They like to trade DNA with one another like baseball cards. So relatedness between different groups of bacteria, it's not going to be a simple family tree. And that's why I wanted to create these phylogenomics webs. Part of why I wanted to create Sequora and get just this idea of how similar are these genomes to one another, rather than strictly talking about things in terms of common ancestry. Because the actual family tree might be very different from what you get in terms of a phylogenomics tree. So if you like what you see here, if you like this tutorial, feel free to go to sequoradna.com, where I've got a whole bunch of bioinformatics tools, and I plan to only build from what I already have. So by the time you watch this video, I'll probably have gone beyond just talking about this pipeline or FASTA merging programs or DNA alignment tools. There's already a whole bunch of tools on sequoradna.com. It is an amazing tool set. However, I plan to only continue expanding it, and I am hoping that if you are watching this video a few months down the road, or a year down the road, or 3 years down the road, I am hoping there's going to be so many tools available, and I hope this will become an industry standard. So please feel free to subscribe, please feel free to comment, please feel free to rate this video, and please share with your friends, share with your colleagues, because I'm just a guy here who is trying to develop, trying to offer my expertise to the scientific community, trying to bring my expertise to the table. And that is what Sequora is. That's what I hope that it will be. I have heard the phrase before. If they don't give you a seat at the chair, if they don't give you a seat at the table, bring a folding chair. And I posed that to an AI, to ChatGPT. And of course, ChatGPT is very positive. We have to take it with a grain of salt. But it said back to me, you're not bringing a folding chair, you're building your own table. And I think in science, we often see things in terms of very Darwinian competition. And we see the natural world and we see things very much through that lens, but we forget that cooperation is also part of the natural world. That is also part of the history of life on this beautiful planet. And so the butterfly helps the flower helps the butterfly. The roots of the trees help one another, and even different species share carbon with one another. So yeah, there is such a thing as natural selection. There is such a thing as different living things competing with one another. That's a thing. That happens. But there is also a lot of cooperation. And I think that as humans, instead of, especially those of us in science, I think instead of thinking of things in terms of zero-sum game, if I win, you lose, if you win, I lose, we should think of things in terms of cooperation. We should be collaborating more with one another in terms of research. So if there's 5 different labs at a university working on very similar projects. Why not work together? Why not get together and combine your experiments so that you can share resources and you don't have to spend as much money? And use platforms like Sequora. This will save you a lot of money. I have so many free tools on there, and by bringing these tools to the table, we're creating a banquet, we're creating a potluck. There's more than enough for everybody if we have an attitude of cooperation and if we want to share with one another. So I hope I'm leaving everybody on a positive note. I know this is a bit of an unusual tutorial. However, I very much intend for this to be the first of many tutorials. So, thank you for taking the time to watch this video. Feel free to check out the Sequora pipeline, the Premier pipeline, and I look forward to seeing you in my other videos. So, shalom and peace out.